the last speaker is Ian Hood. So welcome, Ian. I feel really bad that you've been left right till the end, but it's partly your fault because you didn't want to go earlier anyway. That's good.
No.
And Ian is retired now, but we might well know him from working at Scion, and he's been doing some surveys of fungi on Maungatautari. So I'm going to hand it over to you to share your screen.
Good evening, good afternoon. And as David said, I'm Ian Hood. I'm a forest pathologist with an interest in mycology, and I retired from Scion about 2 years ago, but I still keep in touch with everyone there. I'm still actually casually employed and do odd jobs for them. My talk is about Maungatautari, which is a prominent feature of the Waikato landscape. Here's a picture of it. It's covered in forest with pasture farmland around the base. This shows where it's located here, just in relation to, say, Hamilton, Rotorua over here, Tauranga, and I've now moved away from Rotorua and I'm living near Ahaupo. It's an extinct volcano, about 2 million years old, and it's enclosed in a pest-proof fence, making it into an ecological island. This island is administered by the Maungatautari Ecological Island Trust, a group that reports to Waipa District Council and interacts with 3 local iwi who have connections with the mountain. And the purpose, it's quite a big body, and the purpose that their function is to remove pests, restore the biodiversity, and it's quite a treat to go through behind the fence and hear birds that you don't hear in other places in the Waikato. There's a picture of the fence, and you can also see the type of forest that's on the mountain. Well, I in my retirement, part of the things I've been doing have been looking at fungi on Maungatautari. It's only about half an hour away from where I live, and it's very general exercise, mainly to look at fungi and add their names to the biodiversity inventory of Maungatautari. And in doing so, this corroborates already recorded species and records any additional species. It also allows me to look at what fungi are more common, ones that you see all the time, ones that are less common, and also to relate them to season. In doing this too, I've also looked at interesting ecological observations, and a lot of people are working on Maungatautari through the trust, and they see fungi and they're often very interested in knowing more about them, and so hopefully I can help occasionally with some of their questions. The methods, down on the bottom right here is an aerial view of the area. The white dashed line goes right around the mountain. That's the fence that goes right around the mountain. The yellow dashed line is a subsidiary fence which encloses a sub-enclosure called the southern enclosure. I've picked 5 points which I visit periodically, where I record fungi and collect samples and then examine them macro and microscopically. I've tried to pick sites with woody debris present. And this is the kind of thing, taking photos in situ and then bringing them back, looking under the microscope, doing just recording details about them to help identify them. I'll just get rid of that. I can put that there. The project is very general, as I said, and it has quite a few. It has limitations, not quite a few, but some limitations. But you are getting a biased view of the ecosystem. The first thing is accuracy of identification, and this is, you can do a lot, but it's handicapped by firstly one's own personal knowledge, how much you know about different groups, then the resources that are available. As we know, some literature is much better than others for different groups. I do have to commend commend Bevan on the website, the Landcare website, which is very helpful indeed. Of course, we rely on tools. I'm fortunate to have a microscope. It would be nice to have access to being able to isolate and to have molecular sequencing, but one can't have everything. The other thing is often, as people will know, that sometimes things are missing from when you collect a fruit body. You can't find spores, which hinders your identification. Other factors, I'll just see if I can. I've just got a thing I have to keep moving around. It's blocking the view, but the fruit bodies only, we're just looking at the tip of the iceberg when we look at fruit bodies. For instance, this picture on the right shows a fungus is causing this tree it's decaying out the center, and it'd be nice to know what it is, but if there are no fruit bodies, we can't identify it unless we are able to perhaps isolate or do sequencing. And then by picking 5 sites of fairly small size, that's a good thing, because I can look at the same spots in different times of the year, but it does mean if there are any rare fungi that are only scattered around the mountain, they're liable to be missed. And finally, although I'd hoped to visit on a regular basis, there were various restrictions that prevented that happening. But I think it's still a worthwhile contribution. No knowledge is better than, some knowledge is better than no knowledge. Okay, what results? Well, in the first year, 7 visits were made. Many species were collected. I've identified about 60 species, at least to genus, and many to species. These are some of the Ascomycete species found. 37 species don't appear to have been recorded previously on the mountain, and that's not surprising, of course, but it's good to get these names down. And the fungi found seem to be typical of the sort of thing you'd find around this area and perhaps much of New Zealand in this type of vegetation. And I've been putting collections into the Scion Mycological Herbarium. Here are some of the more fungi from the Basidiomycetes. And I've been creating these little charts to try and get some sort of systematic record. Along the top, you can see the 7 dates, the dates of the 7 visits I've made. I've color coded them as red summer, orange autumn, blue winter, green spring, and then back to summer again, and ticking the little boxes where they have been found, or when they've been found. Here are some examples of the sort of fungi that you will have, that these will be fairly common but to people, but it's nice to see them. Homeomyces volutipes in summer, autumn. Armillaria is in May, June, right now. And winter fruiters quite, these are just examples of winter fruiters. Some, of course, you find throughout the year. Auricularia, or the wood ear fungus, it seems to be always present. There are some exceptions, and perennial fungi, of course, find right through the year. But the question arises: do they have a seasonal sporulation? So I did do a little exercise where I took samples from using a cork borer to take core samples from underneath the pore zone, and then looking at them microscopically. And it's interesting that you do find residual spores present throughout the year. And this seems to tie in with, there was an early study way back in 1984 doing spore trapping in Auckland for the same fungus, and spores were trapped right through the whole year. They did have a diurnal periodicity. So I thought it would be interesting to look at them seasonally. So from 138 collections in the first year, I divided them into seasons percentage-wise. And as you would expect, more are found in the autumn and winter than in spring and summer. So then I went to iNaturalist, and I got the records for Sanctuary Mountain Mangatautri, virtually all the records up to March with 274, and did the same thing. And as you'd expect, I suppose, the same pattern shows up. More records in autumn and winter than spring and summer. I was a little bit suspicious of this very low figure, and it made me wonder whether people know that the time to look is winter and autumn, and therefore that's when they go and look, and so perhaps bias the sampling a little bit. So one of the factors that certain questions arise, and I don't think I'll have any answers, but you begin to wonder, why is some annual and others perennial? Do they have advantages or disadvantages? And similarly, why do some fruit in summer and some in winter? And then what are the environmental triggers? This is a chart of the maximum daily temperature and the minimum daily temperature through the year. You can see the seasons along the bottom. And these are the dates of that I went and sampled. And it's no surprise that as you go from autumn to winter, it gets cooler. But then other things happen too. You get the sun shines less brightly, and it shines less often. As it gets cooler, the relative humidity increases, and you could argue that you get more rainfall in the winter. So which of these is the trigger, or are there more than one trigger? It's probably because they're autocorrelated. It's unlikely you'll be able to find the answers to these questions in the fields. And one day, I guess someone will look more carefully in the laboratory. And climate change is a factor too. As the seasons, as the weather changes, are we going to find changes in fruiting during the year? I did find one interesting reference from Norway where spore size of mushrooms, they found spores were larger if they fruited more in the summer, and they postulated that under the hot and dry conditions, larger spores gave better survival against moisture loss. We've heard about bioluminescence, fungi having sensitivity to or producing light, and I've just talked about sensitivity to other variables. Gravity, of course, is another factor, and this is an interesting one of the sites. This is an old fruit body which obviously formed when this log was standing as a dead snag or as a living tree, but after it's fallen, the new fruit bodies are formed here under the force of gravity. We know why they do that, of course, because the spores need to drop through from the pores or from the gills of mushrooms without hitting the sides. But what the mechanisms are perhaps another factor. So I did look up some stuff, and there's some good reviews in the literature where the whole issue of gravitation is discussed. I mentioned ecological observations earlier, and I thought I'd just touch on some of the niches, the many niches present in the forest. Heart rot fungi are one place, or occupy one habitat inside they're already rotting out the center of the tree, even while it's still alive, usually without harming the tree itself, except perhaps reducing the structural strength. But although there are some pathogens, And then we have endomycorrhizas. If you fossick around the roots of old growth remu trees, for instance, as I did to get this picture, you find these little nodules all along the fine roots of the tree. And it's true of any podocarps, Matai or Miro, you'll find the same. And they house these glomeromycete fungi. Then if we if you push aside some of the forest floor litter, you're bound to find fungal cords or fungal mycelium as they forage. Wood decay fungi they are important for decaying woody debris. This is a very characteristic honeycomb decay that you find in the forest caused by Rigidoporus concrescens. It's the decay is quite common, but the fruiting bodies are very inconspicuous and often not mentioned. These a group of them here. Each of these is like a little disc with a central attachment. They hang beneath the decayed wood, and they tend to their fruit bodies tend to fuse together to form sheets. They have pores underneath. I took these photos just a couple of weeks ago along the vehicle track near the fence. These young fuchsia shrubs, where they're regenerating naturally, were all covered in this native fuchsia rust. Now, just a few more photos, just for your interest, to see some of the fungi found. This one I found recorded in beech forest, but it's definitely also present on Mangatautree. Here's a few ascomycetes. And coming to the end, quite a few fungi, of course, one I haven't been able to identify, so I didn't show those ones, but I thought I'd show one here, which is quite interesting. It's quite common, and you find it most of the year, except I didn't see it in summer. When you squash these little tiny globules, you find conidiophores, verticillate conidiophores with myriads of conidia, but I've no idea what it is, so maybe someone can assist. I'd like to acknowledge quite a few of the people from the Ecological Island Trust have been very helpful and cooperative. Pam Taylor from Sion, who's been putting some of the cultures I've sent, not the cultures, the collections I've sent into our herbarium just as a record. Permit was granted by Waipa District Council to work on the mountain, and also with the cooperation of three of the local iwi groups who have connections. I have done a report. If anyone, I'm happy to share it. If anyone's interested, they can contact me. So thank you very much. I'll see if I can now get back to normal.
Right. Yeah, you should have a red button that says stop sharing up near the top of
yes,
your
I do, yes.
thing. Yeah,
Okay, thank you.
That's it. All right. Fantastic. Thank you, Ian. And what a good retirement project. I'm going to start counting down the days so I can do that sort of stuff too. That's awesome.
Yeah.
Probably the most fungal foray-like talk we've had all day, actually.
Okay.
Okay, well, that's really good. We've got a fascinating project there, a few comments. Oh, Pam Catchaside has got a name for you there, Glucogloria compressor.
Pamela. I'll check that out. That's very good.
This is good. I'm just gonna open this up a little bit. There we go. I've turned off the restrictions, so people should actually now be able to turn their videos on and their microphones if you want to. I don't really mind, but I mean that's basically yeah. Okay. Any more questions there? Really interesting. Oh, yeah, no worries, Kat. Oh, there's Roseland. Hi. Okay. So I've been keeping us all. Oh, yeah, good. Yep. Thanks, Anna. Oh, there's Laura too. Yep. So if there's any other questions for Ian, then you feel free to pop them in the comments or put your hand up, or you can just jump in.
Thanks for that name. I've just written it down to make sure I don't lose it.